Somewhere high in the rainforest canopy, an animal barely moves at all for most of its life, and yet inside its cells, something is happening that scientists have never seen before in any other mammal on Earth. If this sounds interesting to you, hit subscribe right now because we cover discoveries just like this every single day. We often think of sloths as lazy and not particularly exciting, the kind of animal you only really notice when it barely moves at all, but in biology, nothing is ever that simple. It turns out their slowness is not actually a flaw at all. Instead, it seems to be an extremely specialized survival strategy, one that scientists have only recently started to understand, and apparently it comes down to something extreme happening deep inside their genetics. Recently, a team of researchers successfully sequenced the full genome of a species known as the two-toed sloth, and what they found buried inside was something nobody really expected going in. It seems like this animal's extreme, low-energy lifestyle is powered by a strange genetic phenomenon involving what we call jumping genes, along with something that biologists spent decades quietly dismissing as junk DNA, or basically leftover genetic material that scientists assumed was not doing anything important at all. But as we're about to see, that old assumption may not hold up anymore. Before we get into the actual discovery, it probably helps to understand the animal itself a little better first. A sloth is technically part of a group called Xenarthra, which also happens to include some other unusual, slow-moving animals like armadillos and anteaters. Out of pretty much every animal on the planet, sloths seem to have the slowest metabolism of any mammal alive today, often running at around half of what we would normally expect for an animal of their size. That seems to be mostly because of their diet. Since they mostly eat leaves, which provide surprisingly little usable energy, they had to become true masters of conservation, and so they end up spending around 90% of their time completely motionless, barely using any energy at all. But it is not just about what they do, or rather do not do, on the outside. It is also very much about what is quietly happening inside. Their bodies can apparently control their own internal temperature, shifting it along with the surrounding environment in order to save as much energy as possible. This is referred to as heterothermy, which is basically the ability to switch back and forth between being warm-blooded and cold-blooded almost at will, depending on what the moment calls for. And it turns out that pretty much all of this seems to trace back to something happening deep inside their DNA to a degree we have apparently never seen before in any other mammal. Even though sloths barely move at all on the outside, their DNA, it seems, is surprisingly busy underneath the surface. To understand what is going on here, we probably need a few basic concepts first, just so everything makes sense as we go along. We are not really talking about how proteins get built, and we are not even talking about the main DNA strand itself. We are talking about something called transposable elements, or more commonly, jumping genes. These are DNA sequences that can literally change their position along the strand, or even copy themselves and paste that copy into a completely different part of the genome entirely. In other words, they can leave one section of DNA behind and end up somewhere else in the genome altogether. There is a specific type of these elements called a retrotransposon, which apparently works by turning its own DNA into RNA and then converting that back into a fresh piece of DNA that gets inserted somewhere new further along the genome. When this process ends up creating a duplicate copy of an entire gene, biologists call that a retrocopy. For decades, in most animals, these retrocopies did not really seem to do very much of anything useful, which is exactly why textbooks so often labeled this material as junk DNA. It was mostly dismissed and ignored, even though it turned out there was quite a lot of it sitting inside a typical genome, sometimes in surprisingly large quantities. Since many of these copies usually break down over time and stop functioning altogether, there was not a lot of serious research into what purpose, if any, they might actually still serve. But now we are learning that apparently at least some of these genes can become domesticated, meaning the organism somehow finds a clever way to put this genetic junk to use in some surprisingly beneficial ways. This appears to be the very first mammalian example we know of, where junk DNA seems to be doing something genuinely important for the animal that carries it. In this particular study, researchers looked closely at different sloth species, along with several of their closest relatives, and discovered that sloths seem to carry the highest number of these retrocopies of any mammal currently known to science. Most mammals seem to have somewhere between a few hundred and a few thousand of these gene copies scattered through their genome. The two-toed sloth, on the other hand, was found to contain roughly 16,000 of them, with 38 specific retrocopies showing clear signs of being domesticated or essentially repurposed for some kind of ongoing function inside the animal. On top of that, by carefully looking at the mutations built up within these genes, researchers were able to estimate their approximate age and found a fairly clear starting point around 30 million years ago, right around when the ancestors of modern sloths likely first evolved. That suggests this particular genetic strategy has apparently been running for at least 30 million years and seems to have worked out remarkably well for the animal over that time. So, why would an animal go and accumulate so many jumping genes in the first place? And what could they possibly be doing once they are inside the body? By carefully studying the genes located near these retrocopies, researchers noticed a fairly clear pattern emerging. A lot of them seem to be connected somehow to mitochondria and to various metabolic pathways, which suggests they act directly on mitochondria, the tiny structures that generate energy inside every cell. Since mitochondria produce both usable energy and body heat, it looks like some of the original mitochondrial genes changed gradually over time, and that shift allowed these jumping genes to quietly step in and effectively become a kind of control system and backup system all at once. That would mean they are apparently helping regulate mitochondria directly, letting the sloth control exactly how much energy it generates at any given moment, almost like adjusting a dial. In other words, the sloth appears to be using its own junk DNA to build a specialized genetic toolkit that supports its extremely low-energy lifestyle from the inside out. If that still feels a little abstract, it might help to think of it like building with Legos. Imagine you are putting together a Lego set, but instead of just following the instruction manual step by step, you keep finding these random extra pieces sitting in the box that sometimes fit neatly and sometimes clearly do not. In most cases, though, you eventually figure out how to snap those extra pieces onto your model anyway, making it a little more efficient and letting it do something it simply could not do before. In this analogy, those extra spare pieces are the jumping DNA genes, and sloths, it seems, are basically the Lego masters of the animal kingdom. Compared to most other animals, sloths appear to have found a clever way to use these extra copies of junk DNA to help manage their energy use and run their metabolism in a very specific, highly efficient way. So the sloth is not really changing its core DNA blueprint, which still contains all the same original proteins as before, but instead seems to have evolved a way to hack its own DNA by adding in many of these extra copies, each one loaded with its own specialized instructions. This particular use of junk DNA has apparently never been documented in any other animal before, which is part of why researchers found it so surprising. So why should any of this matter to us? On a purely scientific level, this changes how we think about genome evolution. It shows that jumping genes are not just junk or some kind of evolutionary mistake, but can apparently become a primary driver behind an animal's most extreme adaptations. This retrotransposition process seems to be an actual working tool that sloths use to manage their bodies, which also challenges the old idea that this kind of DNA is useless. Even though it does not code for proteins, this study suggests it can still be functional and possibly plays a role in many other animals as well. In sloths specifically, at least 49% of these retrocopies appear to be doing something useful, which means this is clearly not junk DNA after all. But there is also a more practical reason this matters. Many human conditions, including diabetes, muscle wasting, and several neurodegenerative diseases, are often linked to problems with how our bodies manage energy and mitochondrial function. By studying how sloth cells regulate energy and how these animals manage to live in such low energy states without getting sick, we might eventually find biological solutions for some of these human health issues. Researchers might one day even map out how similar retrocopies could be encouraged or domesticated inside human cells, potentially leading to new kinds of medicine. Some of this research could even help us understand how to safely lower human metabolism, which would be useful for something like a long-term space mission if we ever want to travel to Mars without burning through too much energy along the way. By learning how sloths pull this off, humans might one day benefit from similar strategies. There is also a broader, more theoretical idea here about genetic architecture versus genetic adaptation, which acts almost like a counterpoint to the common belief that less genetic material is always more efficient. While many mammals evolved by streamlining their genomes to become more efficient, sloths seem to have done the opposite. Their genome became heavier, loaded down with redundant jumping genes that now appear to help control quite a lot of biological processes. For some specialized animals living in very specific environments, it seems having a messier genome can actually become a major advantage. Being slow and seemingly lazy clearly does not mean being unsuccessful, since sloths have managed to survive for at least 30 million years using these unusual and highly specific genetic adaptations. So it turns out, the secret to being one of the slowest animals on Earth was, quite oddly, having one of the most active and chaotic genomes of any mammal we currently know of. On the outside, everything about the sloth looks calm, unhurried, and almost sleepy, but underneath the surface, it seems like there is a whole lot going on at any given moment. It is a good reminder that in nature, appearances rarely tell the whole story, and it seems there is probably still a lot more waiting to be discovered about how DNA really works, both in sloths and possibly in the rest of us too. For now, though, it is worth just sitting with how strange and quietly brilliant this whole system seems to be.